tube of LV amp and be putting it into here. So what I'm going to do instead is I'm going to show you another technique. I'm going to grab a little ball. So this thing has a little bit of air in there and it has a couple of little buttons. So I want to evacuate the air first. So I press here at the top, I squeeze, and then that lets air out, and then I let go, and I can let go, and this will hold like a little vacuum. When I press this button right here, this S, it will suck air up from the bottom and fill up. And then that will draw up liquid. And then when I want to release the liquid, I press this button here, and then the liquid will draw air from this hole, and the liquid will go down. So we'll just do that method first. So, jam this on the bottom. We've evacuated the air. We're going to give it a quick flame off. Keep everything sterile. And then we're going to open up our tube. We have 8 samples. 8 times 3 is 24. So we'll just go a little bit over 24. Okay, and then this thing is labeled in 2 ways. It counts up from that way, and then down on the other side. And I like to count, which way do I like to count? I like to count down. So I'm going to face this towards me, and I'm just going to be putting 3 mils into each one. So here's 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. And then 2 more. 1, 2, 3. And I need to refill a little bit. So 1, 2, 3. Okay, and I'm just gonna empty this out in the liquid waste. I don't want to contaminate anything potentially. So this, that there. Okay, so now we have our colony PCR all uploaded out, and we have our media. And we're gonna inoculate everything now. So we have our plate that we want to inoculate, and I'm gonna grab my P20. And I'm going to pick up a colony, so a single colony. We're going to do 4 from part 1 and 4 from part 2. So I got a colony, and then what I'm going to do is I'm going to very lightly touch my colony PCR. Just a quick stab, that's it, and eject. If you stab it too much, I've made that mistake before, you're going to mess up the reaction, it's going to throw off everything, and it just won't work. So you just want to give it a very light touch. So 1, 2, 3, pick a colony, give it a light touch, eject into the media. This is number 4, pick a colony, light touch to the colony PCR mixture, eject into there, and now I'm going to switch sides. So we're going to do 5 through 8. So I'm going to pick a colony, give it a light touch, eject into the media. Here's 6, pick a colony, light touch, eject into media. 7, inoculate colony PCR, inoculate the media. And last one, get a colony, inoculate the colony PCR, inoculate the media, and we're done. So now what we have is bacteria in both the colony PCR as well as the media. This is going to go into the incubator, that's going to grow up. This is going to give us a cloudy culture. This is going to go in the thermocycler. It's going to get boiled for about 10 minutes to 95 degrees C. It's going to kill off the cells. It's going to release their plasmid DNA. And then it's going to go into a regular thermocycling program for whatever size part we want to amplify. And that's going to start to amplify the plasmids that we have in that mixture that were released when we killed off our bacteria. But before I go any further, I want to make sure that I label this stuff so that I know what one of these little wells on this Petri strip correspond to what well here. So I'm just going to label out 1 through 8. So 1, 2, 3, 4, 5, 6, 7, 8. And then I'm going to pull out my tips from the block. Grab an air core strip in order to seal off our block. Close it up and label that real quick. So that's going to be 1, 2, 3, 4, 5, 6, 7, 8. Now we're ready to set everything up. Tap this down, make sure that the lids are closed real quick, and we'll get everything all running. So the block is going to go into the shaker. And that goes overnight. And then the PCR mixture, see if this one has the quality PCR program. No. We'll try this one. We have run colony PCR, and we want a 1K program. We have a fragment that's about 700 base pairs, so this will work for us. Just put our strips in there, close it, tighten it, and proceed. Run. We're doing tubes, 25 microliter volume, heated lid, yes. The reason we use a heated lid is if we do not, then the liquid will start to evaporate from the pool, condense up top, and that will throw off the buffering conditions. So we always want to use a heated lid to make sure there's no condensation and all the liquid stays down in the master mix area. So heated lid, yes. And then it's running right now. It'll be done in about 2 hours, hour and 40 minutes. And what this is going to tell us is we're going to run this. We're going to see a band, hopefully of the right size. And then if we see the band of the right size, that means that the culture in there has a plasmid that has a part of the right size. And we can then assume that that is something that we want to look at further. We'll then mini prep it tomorrow, eco-BAM it, map it, and then send it off for sequencing. So that's colony PCR.